Now that we know what cell lines are, how cell lines are prepared and how cell lines are started and cultured, let's look at what they look like. So in the beginning of this, let me show you some example of cell lines that we can grow in culture. Some of these you are actually going to have a chance to look at in molecular and cellular biology lab this semester. My favorite ones by far are fibroblasts, which is right here in this very first image. These are fibroblast cultures. When you look at them, these are long skinny cells and they are growing in these arc-like manner. This is cultured at a very low density or confluency. The density of the cells on the culture plates, which is depicted by what percentage of the surface is covered by cells, is called confluency of cells. So this is a really low confluency culture. So this would be about 10 to 15% confluent, since only about 15% of the cell surface area of the plate surface area is covered with cells. As they become more and more confluent, these fibroblast cultures grow in a very controlled manner. So you get beautiful arcs and turns as they grow, and that's really fun to examine and look at. It's a very organized development of cell culture. Other cells grow in a more haphazard manner. So you'll have single cells that will turn into colonies as they duplicate and make these little cell culture colonies when they are low confluency, as in the case of this epithelial culture. And you can see these are more globular and more irregularly shaped smaller cells. And then here is an endothelial cell culture. These cells can be used to create capillary-like tubes, right? They line all the blood vessels. So these cells, as you can see, are more flat than epithelial cell culture, epithelial cells that you see here. And they are at a higher confluency. So this is about a 75-80% confluent culture. You see very little space left behind underneath. And these cells, when they become confluent, they're all touching each other. So they are beginning to make contacts with each other as well. And the goal at the end of the day is to grow them in single sheets, right? Then we can start to look at their function and see how they behave with each other as well as the environment. And this last one is the second favorite of my cultures, and this is a neuronal culture. So these are cells that are developing into neurons. So you can see the cell body and the different processes coming out of it, mimicking the axons and dendrites that you may look at in the actual brain tissue. We will be doing something like that in the cell differentiation exercise that we are about to start next week, where we are going to be looking at neuroblasts from mouse cells and from mice, and we are going to be treating them with a differentiating agent and looking at how that changes those neuroblasts and starts differentiating them into neuron or neuron-like structures. So that will be a cool observation that you will make from the next exercise, and you will enjoy it, I'm sure. So what do they actually need to grow in this in vitro environment that is outside the normal body system that they are typically found under? So to do that, to think about what they need, how to provide them with that perfect cell culture environment, we need to think about what do cells actually need to grow, to develop inside the body. So obviously, as we talked about earlier, cells need a place to live, to stay, right? They need a home. In many places, they need either a structure or a scaffold or something to hold on to, something to stick to. Inside the body, that scaffolding material comes from those coverings that each organ system is each organ is kind of enclosed in, encased in. And then it also comes from the scaffolding like structure that they get from the extracellular matrix proteins that are making that meshwork that the cells are then going to get attached into. Outside the body, what we do in vitro is that we provide them with that by growing them on flat surfaces. They could be plates, they could be flasks, they could be cover slips or slides, but we make sure that these surfaces that we are growing them on are somehow either coated with positive ions or extracellular matrix like proteins, something for the cells to actually latch onto and grow on. Cells will not typically grow just on a plastic surface by itself. They require that modification to it. So they are usually coated with ECM-like proteins that can then allow them to grow, to attach to them and then grow and culture them. Secondly, they require, obviously, inside our body, cells are not going to be able to grow and continue to stay alive without proper environment, which involves oxygen and the temperature and the pH of the system, and also the nutrients that it must consume, right? We need to have food, we need to have water, we need to have all kinds of things in order to survive. Now, those things are provided artificially to the cell culture that we are going to be developing or using by different medias that we have or different incubators that we have to maintain that environment. So the environment of our cells is going to be maintained by using specific types of incubators, and then nutrients are going to be provided with a chemical concoction of nutrients that we then grow the cells in. And finally, in order to make sure that our technique is good and our data is good that we get from it, we must remain sterile at all times, and we must use aseptic technique, which is something we will learn today in the exercise portion of our lab. We also help to keep our cultures clean and sterile by providing antibiotics, treating them with antibiotics and antimycotics. We live in Florida. Of course, we are going to have spores of mold and fungi in there. We worked with yeast a couple of weeks ago in this lab, so we can have yeast spores around. And then we also have bacterial cultures and things like that that are present in the environment. So we have to make sure that our actual cells are protected from them. So we provide them with all kinds of different things like the antibiotics and antimycotics to help keep them clean and sterile. The media that we use has several different components. The basic media, the base media is actually DMEM or Del-Bucose Modified Eagle Medium, and it is basically Gatorade for cells. So it has the same things that you would normally find in Gatorade. It has water, it has salts, and it has glucose as the energy source. We can also have buffers in there to maintain the pH of the system, and usually you can find it with or without a pH indicator in order for you to have an easy time assessing when your cells need help and need new fresh media. We provide you with media that already has phenol red added to it, which is a pH indicator, and it will allow you to see when your cells are becoming distressed from the pH being either too acidic or too basic. And at that point, you usually need to add fresh media to continue to help them grow. Now that base media is then supplemented with different components depending upon what type of culture you're growing, what type of cells you're growing. It could be that it needs additional keto acids or different types of carbohydrates, glucose versus galactose. You can need to change it to a low versus high sugar content. We can add specific vitamins for specific types of cell culture. We can also add trace elements to them. So several different things can be added to our cell culture. The one thing that we are going to always add to the cell culture, we don't usually add any of these for the cells that we are going to be using in our lab. What we do use are growth factors and hormones to help them grow and develop properly, and antibiotics and antimycotics. The growth factors and hormones are provided to our cells by using fetal calf bovine serum that's really rich in all kinds of different enzymes and growth factors and hormones that will not only aid in the attachment of cells, but will also help them grow and replicate properly. However, you always